Hi, welcome to another week of Soul Seminar Online. Today I'm introducing Janet Sullivan. Janet Sullivan is currently at the University of New Hampshire, Durham at the Department of Science, where she's a herbarium collection manager and assistant professor. Janet received her master's at the University of Connecticut studying the reproductive biology of two aster species. She then continued her studies at the University of Oklahoma studying the systematics of Physalis. Janet's primary research is in the systematics of Physalis with other related genera within Solanaceae. She's co-authored and edited 27 species, which include Aconiki, Petunia, Physalis, Quincula for the Flora of North America Association. Janet has been nothing but an asset to the Solanaceae family, and we're excited to hear her talk on what's next for Physalis. So now you can go ahead and share your screen. Your PowerPoint.
Three-step process.
Perfect. We can see.
Okay, good. Thank you, Stacy. And I do have to say there that I am retired, so I'm no longer the collections manager, but I'm still affiliate faculty at UNH. But thanks for the introduction. And I also want to say right at the beginning that I haven't been on campus in over a year because of COVID. They have a testing regimen that is not available to me because I'm affiliate faculty now, and it just seemed like too big a chance to take to sneak onto campus. So I'm very grateful to all the people who have slides posted online, real high quality images, and I hope that I've attributed all the images properly, and the ones that don't have an attribution on them are mine. Today, what I want to do is introduce the genus Physalis, because we've had a lot of talks on Solanum and other genera in the Solanaceae over this past year, and I'm not sure that people are familiar with the genus. And I want to summarize the research to date, and then talk a little bit about where I think we might go with study of the genus in the future. Oops. Oh, interesting. Now my slides are not advancing.
Click on the slide.
So sharing.
Click on the slide.
On the slide. Oh, got it. Okay. Thank you. My husband and I are parallel playing on our laptops in our dining room office. So the genus Physalis is an American genus. About 90 species, and I realized as I was writing that I've been saying that for about 35 or 40 years, and there are species that have been taken out of the genus and described in other genera. New species have been described. It's an estimate. I still think it's valuable to estimate at about 90 species in the genus. About 2/3 of those are in Mexico. There are North American, north of Mexico species, and throughout Central America and South America, but predominantly a Mexican genus. It's recognizable by this inflated fruiting calyx, that classic bladder, which the name Physalis comes from. It's Greek for bladder or bellows, and probably familiar with the winter cherry or the Chinese lantern plant, which is no longer in the genus Physalis, but it's that distinctive inflated fruiting calyx. The best image of the open fruiting calyx I found on Etsy, just surfing around looking for images. And so you can see the berry inside of that balloon that expands out. There are some species that hug a little bit tighter around the berry, but for the most part most of the species form this nice big balloon around the berry. Most of the species in the typical species in the genus have yellow corollas, either yellow or cream yellow. They're pendent so hanging down, or if not fully pendant, then nodding. The flower in the lower right-hand corner is propped up on a leaf so that you can see the markings in the throat of the corolla. Sometimes these are really distinct and they're very dark, sort of purple black or dark brown, like you see here. Sometimes they're more like a smudge. I liked that description of Maggie Whitson's, that they're just like a little smudge of color, sometimes yellow or green or sort of a tan ochre color in the throat of the corolla. And the flowers are borne singly at the node in the typical species in the genus. So that inflated fruiting calyx, the pendant flowers singly at the node for the most part, most of the species, and then pendant or nodding. That inflated fruiting calyx, I wanted to say here too that, and I think this might have been mentioned before in Rocio's talk, that I think it's an important dispersal mechanism, even though the fruit itself is a berry, which we think of as being dispersed by animals, eaten, and then the seeds pooped out somewhere else. The inflated fruiting calyx encloses that berry, and it's really very buoyant. I've seen them floating on down streams or small rivers. I've seen them blowing along roadsides and sand dunes. So wind dispersed or water dispersed, I think that inflated fruiting calyx plays an important part in the dispersal mechanism of the genus, of members of the genus. So even though we have this fairly uniform description of what the typical members of the genus look like, once you get to know them, there's really a lot of variability. There are annual species, perennial species. You can see in this slide just a lot of variation in the shape of the leaves, from elliptical to more lanceolate, entire margins, toothed or lobed margins. The one in the upper right is more of a deltoid shape. Some of them are very narrow and strap-like. And hairs are very important in distinguishing species in the, as a vegetative character. And you can see also some of the variability in the flowers here too. The one Sandy's picture down there in the lower right is one of the annual species, and a little deceiving, but those flowers are pretty small. And then in the perennial species, for the most part, the flowers are bigger. The length of the pedicels varies among species. You can see some of the different shapes of the corollas. So campanulate-rotate is the typical term that's used to describe the corollas, but you can see the one in the upper left-hand corner there is quite widely flaring, much more rotate than the one below it, which looks like a nice swirling skirt. And then for some species, the limb of the corolla reflexes when the flower is fully open, and that can be distinctive also. And those features can all vary within a population as well. And then the fruits in the center, the two different forms really. The lower one you can see is really very sharply 5-angled fruiting calyx. And the one in the upper central image, you can see those 5 prominent ribs, but there's also another prominent rib in between those. So really more round in cross section. And I call those 10-ribbed, so 5-ribbed or 10-ribbed. And that's something that and the floral morphology are things that once specimens are pressed and you're seeing them in the herbarium, they can be very difficult to discern. Even annual or perennial, a lot of people will describe a species as annual when it's actually perennial and vice versa. And the same with that fruiting calyx, which to me is so distinctively either 5-angled or 10-angled, even when it's pressed. But of course, I've looked at a lot of specimens. So when you're not familiar with all the diversity and you haven't looked at a lot of specimens, you can really lose a lot of distinguishing characteristics. And so sometimes they're very difficult to identify. Economically, the family is important. The one everybody's probably most familiar with is the tomatillo, which is used in salsa. And that's a savory, a tangy fruit. It has a little bit of a bite to a little bit of an acidic taste to it. The rest of the genus are all really quite sweet, and you might be familiar with the Cape gooseberry or the strawberry tomato that are used in pies or jams or as a sauce. Or I love this picture of the garnish. A friend went to Scotland, and that's haggis with Physalis on it. So she said, "Look what I have met in Scotland." And then again, that Etsy picture, because I've also been in restaurants where I've had Physalis as a garnish on top of a dish, but I've had it where the calyx gets turned over so that you get that sort of star of the torn fruiting calyx with the berries standing up there in the middle. I think that's very attractive. And in the literature, they're mentioned, Physalis purviana and Physalis minima are mentioned as being used for antispasmodic or diuretic purposes, as an antiseptic, a sedative, and analgesic. And of course, if you go on the internet, you can find all kinds of interesting information. So when I was surfing around and looking for pictures, I found recipes for Physalis martinis and margaritas and crepes and curries, or fruits used in a tea or as a dried fruit in raisins and peanuts mix or whatever, or even used to sweeten salsa, because of there's a lot of variability in salsa now too, with things like mango and pineapple. And then also health benefits, and I'm not sure, I put a question mark next to this, that Physalis is mentioned to help improve vision and aid in weight loss and lower cholesterol and blood pressure and aid in Parkinson's disease, as well as protect against colds and flu. So that's kind of interesting, and I don't think any of that's really substantiated. And then also, like other Solanaceae, remember that the foliage and the unripe fruits can have solanine and solanidine alkaloids in them, and so would be toxic in large doses. And there are mentions in the literature and on herbarium specimens about poisoning of livestock. Physalis was described first by Linnaeus in Species Plantarum in 1753. He listed 9 species in the genus. And I should have mentioned too, an American genus, but that there are species that have been distributed worldwide. And so by 1830s, Nies had enough species and was confused enough that he published a paper which a title translates to attempt to understand species in the genus Physalis. I thought that was interesting. So even 80 years later, there are some species put into synonymy, some new species described, and taxonomists are confused. By 1875, Asa Gray, who had a lot of specimens at his disposal because a lot of people were collecting and sending specimens back to him at Harvard, and he started his synopsis of the species of Physalis by saying the North American flora can hardly contain a more difficult genus for its size. Part of that might have been that he included species in the genus that we now consider to be different genera. So if you're familiar with Camasoreca or Quincula or Leucophysalis, and that would certainly make for a very polymorphic genus and maybe difficult to figure out. He listed 17 species that he said were in that typical group, ones that are now considered more typical Physalis and still in the genus Physalis, but his focus wasn't Mexican species. And so he's really missing out on a lot of the diversity in the genus. In the 1890s, Rydberg published a synopsis of the North American species of Physalis. And again, his focus is not on Mexico. It's mostly on specimens in the US, some Canada, and some Mexico, where the species ranges extend down into Mexico. And he started his paper by saying a revision of the genus Physalis is without a doubt much needed. So 150 years after Linnaeus, people still haven't figured things out. One of the things that he did that was very helpful was that he divided the genus into species groups or series, and those were the species groups that we used for the next 100 years to help get a handle on the diversity and to work with the diversity in the genus. In the 1950s, the first person to do something other than look at morphology was Margaret Menzel. She was actually a cytogeneticist. I'm not sure what that yellow line came from. She published a paper. She actually published a series of papers, but the big one is the cytotaxonomy and genetics of Physalis. So 200 years after Linnaeus described the genus is when we're first getting a different approach to trying to understand the diversity. She looked at the cytological characteristics, but she also did crossing experiments among different species within the genus. She worked with what she could identify as 25 species and then several of what she called doubtful forms. Some of them just say, have a question mark after them in her paper, or just say species 1, species 2. She'd already reported on the cytology in Camassarica, which she recognized as distinct, and Leucophysalis, which wasn't always recognized as a genus distinct from Camassarica. She considered Quincula to be distinct from the rest of Physalis. And the genus Margaranthus, and you'll meet all these with pictures in just a minute. So some of the, actually, one of the important things in the very beginning of her paper, she starts off by saying, since the description of the genus by Linnaeus in 1753, species determination in Physalis has been in a state of confusion. This has been due in part to faulty description and misapplication of species nomenclature, and in part to the fact that most of the members of the genus are morphologically rather similar. Once you take out things like Leucophysalis and Margaranthus and Quincula, you get a much more cohesive genus. She still had some things that she thought might not be best treated within Physalis, but this was what she worked with. She confirmed that the diploid chromosome number is 24, and that there were tetraploids of Physalis angulata and peruviana, so diploids and tetraploids for both of those species. And she concluded that polyploidy is not an important factor in the evolution of the genus. So some good ground rules here, or background information on the cytogenetics of the genus. She, as I said, did crossing experiments, and she found that there were barriers to hybridizations among the annual species, but not the perennials, which is kind of interesting because when I look at the list of what she has as separate species, some of those things are now synonyms. So it's interesting that even in the annuals, she didn't get hybridization. And she also noted that there were 2 species, what was known at the time as Physalis carpenteri and Physalis alcakengi, that Chinese lantern plant or winter cherry, that had what she called more primitive features, well, now we would say ancestral features, chromosome features in the genus. Following her work later in the 50s and then into the 1960s, UT Waterfall monographed the genus in 2 different stages. First, the North American north of Mexico, and then he tackled the Mexican, Central American, and West Indies species. And what we see with both of these researchers is really that they're starting to wrestle more seriously with generic circumscription. What's interesting, though, is that Waterfall didn't adopt some of the cytological conclusions about relationships that Menzel had come to based on cytology and crossing experiments. And so he still really kept with that, what I think, that inflated fruiting calyx, so nicely illustrated here in this lower right-hand corner, where you see that calyx is really ballooned out. It's really widely inflated around the berry. Whereas some of the other things, Chamaesyrica and Leucophysalis, which he included in the genus Chamaesyrica still, that inflated fruiting calyx is much more tightly invested, so very snug around the berry, and the berry sort of peeks out of the tip of it. And so that, I think, seemed to be important for both of them in identifying this cohesive genus Physalis. And one of the things that we've learned recently, and I'll talk about this a little later from the work that Rocio has done in Stacy's lab, is that accrescent calyces have arisen more than once in the Solanaceae. And so here they were relying on this very distinctive characteristic that might not have been such a good one to rely on, but it's kind of hard when you're looking at a lot of herbarium specimens and a lot of the features are kind of disappearing into this flat specimen, and you can't really see some of the other differences. And of course, they didn't have all of the tools and techniques that we have now. And then the other thing that I think was really important for these researchers was that pendent yellow campanulate-rotate corolla, although there were exceptions to that, and I'll talk about those here in a minute. And Menzel, in her work, mentioned maybe this group of species doesn't belong in the genus, and Waterfall had, he had more of a broad concept of Physalis, and he was saying maybe things like Chamaesyrica really belong in Physalis. Maybe we should have this big polymorphic genus. So that really, as I say, starting to wrestle with that generic circumscription. So some of the misfits, what I call them misfits, and I mean that fondly because I think these are just such great plants. On the left here, what's now known as Alcakengi officinarum at the time was in the genus Physalis, and actually it was the type of the genus until Maggie Whitson retypified it based on her dissertation work. It's really, everybody said, gosh, I have no idea why I keep getting these scribbles at the bottom of the screen. Anyway, she, so Alcakengi is very different. Everybody would remark on that in any paper about Physalis, would say how uniform the members of the genus were, except there's this thing that has white flowers with a lobed corolla, and it's Eurasian, it's not a New World. So things that made it really distinctive and really different. One of the other things is Calyphysalis, now Calyphysalis carpenteri, which was known as Physalis carpenteri until, again, Maggie described this new genus. And you can see why it would be included in Physalis. I don't have the fruiting calyces there, but it does have that inflated fruiting calyx. It has pendant yellow flowers. You can see in this picture, though, that there are multiple flowers per node, so that's a little different. The other thing is that it has these sclerotic granules, which we've heard about in Solanum, and they also show up in Lisianthes, that we don't find in any other Physalis. So those little brown seeds at the bottom, and then those sort of tan round balls are the sclerotic granules. So that was something that these two species were always things that would get pulled out right away at the beginning of any key because they were so distinctive and so different. Oh, and remember too, that Menzel, these were the two taxa that Menzel had said had a more primitive cytological profile than the other members of the genus. So things that just add to that list of characteristics that make them really different from the typical Physalis. And now they're recognized as distinct genera. Some other misfits are the genus Celtalia. They're on the left-hand side, described in the 1990s. This is one of the things that Menzel had called out. At the time, it was 7 species that were had more of a shrubby habit and had multiple flowers at each node. And so she had said, maybe these don't really belong in the genus. And sure enough, described in the 1990s as a distinct genus based on that. Not all of them, not all, 2 of the species, actually 4 of the names that Menzel had mentioned became the 2 species in the genus Celtalia based on that shrubby habit, fasciculate flowers, so more than 1 flower at each node. So you can see they're very distinctively lobed corolla, which is not what we see in the rest of Physalis. And then a couple of other features, pollen features, and the fruiting calyx is more coriaceous. And then the genus Quincula, which really is so distinctively different. It's you can see different color than the rest of the genus. The hairs, I'll talk about in a little bit, but you can see what look like little white grains of sand on the leaves there are actually hairs. They're like little balloony type hairs. It has a different chromosome number, base number of 11, really very different than the rest of the typical Physalis. But these were all things that Waterfall, those 4 genera were things that Waterfall still kept retained in the genus Physalis. Interestingly, things that he maintained as distinct, Chamaesyraca up there in the upper right. And I mentioned the fruiting calyx isn't balloony inflated, but it is expanded, a crescent, and just tightly hugs the berry. And then Leucophysalis, which he considered still as within Chamaesyraca. And the other thing that's been just a strange little plant is this, it was for a long time Margaranthus, a separate genus Margaranthus. And you can see why, even though it has that inflated fruiting calyx, that balloon, distinctive balloon or bellows, the corolla doesn't look anything like any of the other Physalis. It's got that eye-popping purple color. It's urceolate. It's not anywhere near campanulate-rotate or a reflexed bell or anything. It's just so very different. But you'll see in a little bit in the 1990s, once molecular techniques started to be applied to the genus, it falls right within the typical Physalis. So making people like me who have to write a description very unhappy. So now coming into the 1970s, into an era when people were realizing we want to apply other techniques to understand relationships and among species across the angiosperms. And this was termed biosystematics. And the first person to apply biosystematic techniques to Physalis was Fred Hinton. And he did a lot of work very helpfully, ground level, knowing the species in the field, but also doing flavonoid chemistry, analyzing the flavonoid constituents, crossing experiments. And he helped us understand that Rydberg's series Lanceolata, that Physalis lanceolata really was a species of the Carolina sandhills, not something that was widespread in the central plain states of the US. This Physalis pumila and Physalis hispida, he considered conspecific, and that they were distinct from Physalis virginiana. So a lot of the specimens, I still see a lot of specimens that have confused nomenclature because of not having gone back to these papers and looked at his descriptions of these species and seeing how different they really are, how distinctive they really are. And there you can see that nice hispid pubescence on this Physalis pumila, what's now variety Hispida. So when I came along into graduate school in the 1980s, I wanted to similarly apply a variety of techniques to an understanding of Physalis. One of the things that also that I was very interested in was that capability, the crossing capability. So one of the things that I did was to examine pubescence in the genus, and I chose one of a manageable size, because if you're going to be doing a variety of analyses, you can't look at 90 species. You really have to get a more manageable subgroup of that. And so I chose Rydberg stellati, the species that had these dendroid stelliform hairs. There on the right, you can see really complex branching. And actually this part was really not my thesis work. It was done in connection with a woman in Germany who very patiently, I sent her specimens and I sent her seeds, and she very patiently grew these things on her windowsill and then drew the hairs, very painstakingly drew the hairs. She was familiar with solanum. She'd worked with solanum hair morphology before. And so these, and I'll use my cursor as a pointer here, these, what she called storied glands, these little short storied glands show up in solanum, and they also show up in Physalis. But otherwise, the hairs were very different. There were branched hairs, as you can see, branched hairs and unbranched hairs, and I've shown these on different, well, the branched ones don't show up so nicely on photos of plants. They show up better in electron microscopy, but you can see that really complex branching and different from the stellate hairs that we tend to find in solanum, where all the branches come out of one point. They really form these very complex trees, and it was very distinctive for the group that I worked on. And then since then, with the molecular techniques, it's been shown that thankfully, that was a that is a monophyletic group. So so I got a good group to work with, and working with Amit Saitha, she showed two trends in developmental trends in Physalis pubescens. One is developing from unbranched hairs to branched hairs. So you can see down there on the bottom, the simple hairs that developmentally branch and then get the much more complex branching. And then a developmental trend from glandular hairs to glandless hairs. So sometimes young hairs glandular, and then as the developmentally as the plants mature, developing glandless hairs without glands. And you can see too the simple hairs. I sent her not just the members of Stellati, but members of other series, others of Rydberg series too, to get a perspective on the group that I was working with. And we concluded that there were differences among species and among series within the genus. We don't recognize those same series anymore, but that those species groups were distinctive in terms of their hair morphology. And then I put the quincula down here at the lower left-hand corner again, so that you could see those really they look like little beads. They look like soil particles or sand or something, but those are hairs, and they're like these little balloons. They're like little sacs, or they've been called vesicles. And as they mature, they sort of split along the base and kind of pop up. So like a burst balloon that hasn't collapsed, but really very different than the hairs in anything else in the genus or in the family, I think, as far as I know. So the hair morphology was important developmentally and in terms of systematics. I also wanted to examine the pollen, partly because I was at a really good EM facility in Oklahoma. And some of my pollen images are on the left-hand side, one of the plates out of a publication. And those are, I think those are 4,200, magnified 4,200 times. And when I expanded to 15,000 X, you could see that those little bumps are really little spines, really little pointy bumps, I guess is the way to say it. So the term that we used to describe that was microspinulate. And there were some differences. You can see some of the extensions over the culpus or the furrow there. Some of them are pointy, some of them are sort of rounded and curved. You can see slightly different shapes, but really the pollen in the genus, as it is in the whole family, is fairly uniform, and so wasn't very informative in terms of distinguishing species or distinguishing series within Physalis. In the lower right, those lower two images were quincula that we did also, and that really did have difference, a very different shape, and the pollen grains were actually very large compared to Physalis. So maybe among genera, but still kind of marginal value systematically. And then when they described the genus Celtalia, Estrada and Martinez also examined pollen, and they described it in the genus, in the two species that they described in that genus as a scabrate, and different from the granular exine that they saw in another one of the woody or shrubby Physalis. I think one of the things that Menzel had called out as something that maybe didn't belong in Physalis, a more shrubby genus with multiple flowers at each node. So one of the things that I really wanted to do, I was interested in pollination biology, and because I thought, you know, it's all well and good to know that things have the potential to hybridize, but if it's not actually happening in the field, then you know, what good is that? I mean, I guess there's value in knowing that, but I thought the pollination was interesting. So I conducted a pollination study of what was then variety cineracins, now Physalis cinerat, back to being it's recognized at the species level. And the populations that I studied had Physalis cineracins, but my primary populations, but I also studied other populations where there were other sympatric species, Heterophylla and Longifolia and Pumila from other sections in the genus, just to get an idea of, is there a difference in pollination syndrome? And you know, here you have these flowers that across the genus are so very similar, and it's an interesting pollination syndrome because the flowers are pendent. They're not sticking up out of the leaves so that they can be seen from any kind of distance. There's no fragrance. And what I found is that there was really a uniform pollination system across the genus. I've got pictures of the 3 bees that I found on members of the genus. And actually, that perdita, the one in the upper right-hand corner, had been collected by entomologists on Physalis species and other places in the US, which was kind of interesting. The common name of it is the ground cherry fairy bee. So it is the Physalis bee. But I also found 2 other, Corollas, the green one, and Calyx latitarsis, all visiting Physalis. They all collected pollen. The perdita also did probe for nectar, and you can see those big whitish yellow things here with the cursor. These are the blobs of pollen on the bee's legs. They form these very hard packs of pollen. They collect the pollen and they go off and sit on a leaf or the top of the back of a flower, and they groom themselves, and then they pack the pollen into these pollen baskets, their scopy, and bring back to the nest. And I did find that perdita carried 100% Physalis pollen, And the Calyx that I collected did also. Auga chlorella was a little bit more of a polylectic bee, and I but usually found more than 60% pollen on the insects that I collected. So where cross-compatible species occur sympatrically, hybridization can occur. And Hinton had documented that in the Lanceolati group. And then I also found that these are names from Waterfall's names. So there are variety names, or he sometimes just said things were intergrades. You can see down there in the lower right between Mollis and Variovestida. And so I had done a lot of crossing experiments in the greenhouse, and the cross compatibility, the thickness of the lines indicates the degree of cross compatibility, and the thickness of the lines corresponded with other features that I was studying. And so the circles represent either species, or in the lower left there, a species group where hybridization has occurred between what used to be variety meridema is now Physalis walteri and Physalis angustifolia, and forms these variety Eliadii, variety glabra intermediates in Florida. So everything changed in the 1990s. Morphological data information started to be combined with molecular analysis, and it became really important in understanding not only the circumscription of the family, but phylogenetic relationships within the family. And some people that are here today have contributed to that. And I don't know everybody who's here, but I did see Lynn's name pop up, and maybe Tina's here, or Dick Olmsted, among other people. So a lot of what we know now about the circumscription of Physalis, as well as the circumscription of the family, has come out of that research. And some big things that have happened, and I'll start with the Whitson and Manos over here on the right, the 2005, just because Maggie focused on Physalis and physaloid genera, other things that have inflated fruiting calyxes. One of the things that came out before her work, actually in the 1990s, was this Margaranthus. Remember that little thing with the eye-popping purple urceolate corollas, but an inflated fruiting calyx. And so here it is coming out consistently in her study within the morphologically typical Physalis. And then that is also not illustrated in here, but it also comes out here in the 2016 study. So, what do we do with that? And the other thing here is quincula consistently falls out here, separate from the morphologically typical Physalis. And here identified as Physalis lobata in this study, but really not coming anywhere near the more typical Physalis. So if we're going to include quincula in Physalis, we really have to have a much more polymorphic genus. I should say too that Whitson and Manos did their study using Waxy and ITS, and the Zamora Tavares et al. Used those same 2 genes and 5 plastid genes. But what we get consistently, you see little differences here and there, but there's Celtalia, very different from the typical Physalis. There's Leucophysalis, very separate from any of the Chamaesyraca species. One of the things that came out of Maggie's thesis work here is what had been Physalis alcakengi and Physalis carpenteri, really very different. If you're going to include them in Physalis, then you also have to include Chamaesyraca, Leucophysalis, this weird little Oryctes monotypic thing in the Southwest. And so she, based on this, went ahead and named the new genus Caliphysalis for Caliphysalis carpenteri. and then retypified the genus so that Alka kingi is a separate genus also. But one of the things that we can see out of both of these confirmed again and again is that the subtribe is monophyletic, the subgenus, the typical subgenus Rydbergus is monophyletic. One of the things also, though, is that everybody has said that there's poor resolution at the species level in some cases. So the molecular data is not going to tell us the whole truth. We need to use it as a tool combined with other tools. Just to wrap up here with more recent paper, this Zamora Tavares again studied recent divergence or showed a recent divergence among species as had been suggested by Whitson and Manos and by Olmsted et al.'s work. So, but they put a clock on it here, which is interesting in understanding that divergence and distribution of members of the genus. Everybody points out that there are morphological changes in places where there haven't been genetic changes. So, and using that as evidence that possibly, or using that as evidence that there's probably recent among some species. And then I wanted to mention Rosio's work here about the inflated fruiting calyx, which she spoke about in this seminar series a while ago, showing that those that inflated fruiting calyx is involved many times. So maybe it's not. That would help us understand the phylogeny and help us not to latch onto that quite so much as a defining character, that we really have to use that as one of several characters to identify different groups and see that it pops up different places in the phylogeny of this subfamily. And this up here in the upper right, this is that Oryctes, that weird little thing that pops up in the southwestern US. And you can see very different corolla shape and expanded accrescent calyx, that it's the lobes that expand in fruit, but the fruit is not enclosed in that balloon, or that berry is not enclosed in a bellows or a bladder. So there's still a lot to be done, lots to do at the genus level. There's this little clade here that I'm pointing to with my cursor. These are two of the taxa that Menzel had called out as being things that maybe didn't belong in Physalis, more of a shrubby habit and sometimes more than one flower at a node. And I'm not familiar with those. They're Mexican taxa. And but I just think it's interesting that she called that out based on cytology and combining that with morphological information. And then here they are with molecular data coming out separate from the typical Physalis. And I seem to have lost, oh, there it is, my cursor. Physalis low beta, really distinctive. I don't know why it's still called Physalis and not quincula, because it really is very different. But I think it's interesting that here it comes out as closely related to Physalis microphysa, again, which I don't know, that's a Mexican taxon. And so I think those are things that deserve a little bit more study. But then there's so much still to do at the species level. And again, I'm really most familiar. I've done some field work in Mexico, but not a lot. I'm most familiar with the US taxa. But one of the things that I see again and again is that field knowledge is so important. A lot of times herbarium labels don't give you all the information you need about ecology, about sympatric species, and you can't always get an idea for how the plant is holding itself, how the the flowers are held on the plant. And so it really is important to have that field knowledge. One of the newly described species is this Physalis macrophysa, just described within the past year by Milo Pine and colleagues. And it's something that he had his eye on for a while. He described it as a distinct species based on a larger obpyramidal fruiting calyx and large seeds and a coarsely pitted seed testa. It's something that I would look at an herbarium specimen and say, this looks an awful lot like Physalis heterophylla to me. Physalis heterophylla is a wide-ranging geographically, it's variable, but he knows this group in the field, has spent more time looking at these in the field, and he really thinks that this is a distinct species and described it as such. He also mentioned this samophyte characteristic. That's I had to look that up. It's a term meaning adapted to living in shifting sands or sandy habitats. But the plants that I worked on grew in sand dunes in Florida and up the East Coast and down into the Yucatan and sand dunes. And on sandy roadsides, the more inland species were sandy roadsides. And so I'm not sure that Physalis pumila subspecies hispida, variety hispida, occurs in the sand hills in the central part of the U.S. in the plain states. But I think that's interesting. I think, I'd like to hear more about that from him. So there might be others, if this is really a different species, maybe there are other variations on Physalis heterophylla that I wasn't recognizing as distinct species or varieties that are. Also Physalis heterofolia, sorry, H-E-D-E-R, excluding Physalis fendleri, which is really very different. That's a southwestern group. And again, lots of different species described that once you start looking at a lot of herbarium specimens, really, I don't see distinctive groups, but I haven't done as much work in the Western plain states and the Southwest as I did in the Southeast. The Physalis crassifolia complex is under study, and that's, again, Southwestern, Southern California, Arizona, and south into Mexico. And again, field botanists see differences there, whereas looking at herbarium specimens, I had trouble identifying distinct species that I could recognize reliably. I think climate change and habitat destruction are going to be important because they'll have ecological impact on a lot of these taxa, especially the ones that are growing in areas. Every time there's a hurricane, I think, oh, what about all those populations I visited along the coast in Florida and Gulf Coast states and down into Mexico. So I think that that form of habitat destruction is going to be important for coastal species. So I think there's a lot to do still, and I think that I'd like to hear from other people what they think, what they know about Physalis and what they, where they think we should go from here. Thanks for your time. Thanks for the platform. Thanks to the organizers for inviting me. And I look forward to questions.
Awesome. Thank you so much, Janet. That was great. It was a great overview of Physalis and what's been done so far. We now can open for questions, so you can type it in. You can mention you have a question. Eric asked a question in the beginning of the seminar about interspecific crosses. Maybe he wants to add on that, but I think you addressed some of that with your work. But if not, you can always unmute yourself and ask it more detail, Eric, if you want. But Stacy has a question, so if you want to go ahead, Stacy.
Okay, Stacy.
Yeah, That was really great, Janet. I'm sorry if I put little reactions in there that were distracting. You cracked me up with some of the things about like how hard Physalis is. I regard it as one of these, you know, fascinating genera that just boggles my mind, you know, that that there's so many species, but yet the phylogenies all differ. There's all this conflict. There's within the species, so many of them are so variable. So it just seems like there's so many points to work on. So I've been really happy that Chelsea has been tackling some of it, because we just clearly need lots of folks working on lots of different aspects. So I was wondering about, so you mentioned, you know, how many of these are cultivated, and I've taken to tasting lots of them. It seems to me that many of them are edible, even if they're never cultivated. Has anyone ever done sort of a survey of this? And should I not be putting them in my mouth?
No, I do. I don't eat the green ones. I'm never sure about the ones that are green. You know, so I wait until they ripen up. But everybody doesn't like them, and it's interesting that, you know, my family is like, oh yeah, they're okay, but you know, I'm like unwrapping them like candy. So they never make it into jam or pie at my house because I just eat them all. And it was interesting. I listened to Mahinda's talk a year ago or so, and she was saying that, boy, those sweet species just don't catch on. That people like the tomatillo, they like that tang, that acidic tang, but the sweet species don't really catch on. But it so let's see, what was your question? So yeah, go ahead and eat them, and I like them. And oh, you did ask about not cultivated, but they are gathered in the, especially, you know, prairie states, I think, where you can find large populations, and they are mentioned as being used in pie or in jam. And sometimes I'll see on herbarium specimens that they've been used, been collected and delicious or whatever.
It would be interesting to see about the color association, because you're right. I think I tasted someone who was growing in the sand dunes in Florida. Maybe it was Arnicola, and it was pretty tasty and it was bright yellow. But then I tasted the green fruits of acutifolia that Chelsea has in the greenhouse, and they're terrible. And so I wonder if there is a green yellow thing. That's gonna be my new Physalis little side investigation.
I know there's so much to do. Yeah, so much to do.
Well, I can help by just tasting. I have to run to something else. Thank you so much, Janet.
Thank you.
Awesome. So Eric was just asking similar thing. Are they all edible, or there's some toxic fruit? And I don't think there's any toxic in like the crown Physalis, right?
I haven't heard of that. The only thing that I've seen is livestock being poisoned or not well after grazing in a field where there's Physalis. And maybe it's, you know, green Solanaceae just seems like something we should stay away from, you know. Although tomatillos, I mean, some of them purple up or, you know, get that yellowish and purple spotted look to them, and they're fine. So, and people eat green tomatoes. So
it's kind of a hot topic, but do you think there's a difference between Physalis philadelphica and Physalis iocarpa, the purple?
Oh, Ixocarpa?
Yeah, Ixocarpa.
Yeah, oh yeah, Don Hudson, actually, that work had been done, he mentions it in his, he worked with Physalis philadelphica, the wild and the cultivated, you know that. And he refers to the Ixocarpa work, and they really, you know, once you look at a million herbarium specimens, you can tell the difference. Philadelphica really has much larger flowers, and Philadelphica, I always think if I look at it and I think, oh, that looks like Physalis lanceolata, except it has really short pedicels, then I think, oh yeah, right, it's Philadelphica, which isn't native up here, but you find populations that have become naturalized. And Ixocarpa, the flowers are really very tiny, very different. But
do you think that's like due with selfing? Do you think iocarpa is selfing and Philadelphica tends to be outcrossing?
Oh, do I? That's your area.
Yeah, that's true. That's true.
You can answer that question.
Maybe. Okay. Pilar also said that Mexico has all tasty species, and some are sweet at first and bitter at the end, but none are toxic.
So even green ones,
Pilar, do you want to unmute yourself?
Do you eat green ones too?
Yeah, there we go.
Hi.
Hi.
Aquí, ¿puedo decirlo en español? Porque me cuesta.
Okay.
Maybe, Rocio, can you help?
Por favor.
Yeah, sure, I can help.
Los verdes incluso los hemos probado y no son muy, pues no saben bien. Entonces, más bien, no los comemos porque no saben bien, pero no son tóxicos.
So what she's saying is that the green fruits, they have tried them and they are not taste very well. So they don't eat them. Like usually they are not eaten because they are not very tasty, but they are not toxic at all.
Okay. All right. That's good to know.
Tengo otro comentario.
¿Sí?
Okay.
Solo que, bueno, ya estamos a punto de someter por fin vamos a someter el trabajo de microfisa y se va a llamar Microphysalis dehiscens. Ya Cascada no logramos completar el nombre. Y también coincido en que Physalodendron, todo lo que es un género Physalodendron, debería de estar en otro género. Y también vamos a trabajar en esa parte taxonómica para que sea un nuevo género. Entonces serían 15 géneros ya en Physalidines, ¿no?
So what she's saying is that they are about to submit this work that is the Physalis microphysa that is going to be called, how do you say?
Microphysalis dehiscens.
Microphysalis dehiscens. And they're also working in the Physalodendron clay that it's going to be like a different genus as well. So at the end, it seems that there are going to be 15 genera in the Physalidines subtribe within Physalidae.
Okay, good. Yeah, I thought that those two species had already been described as a separate genus. So the two shrubby ones. So, and then when I started to surf around, I thought, no, they're still Physalis. So good to know that.
Watching it work.
Yeah.
Sorry.
It's like
I was
Eric.
going to ask, like, if Physalodendron probably is going to retain that name because it was a section probably before, and yes, that would be easier to identify that way. Probably.
Yeah.
Eric also has a question about one of your slides showed geographical distribution. Is Physalis global distributed? Is it the center of origin North America?
Well, it looks like from that 2016 paper, really that Mexico is, right? I can go back to share screen and, oops. Okay, did I get that?
We can see your thank you slide.
Okay, let's see if we can go back. Oh, I'm sorry. You know, I've read so many papers over the past couple of weeks again. So, but down here into North America, including Mexico, I think, and then spread from there. And that there are species that have been introduced outside of the Americas. And I get requests to identify things all the time. And usually I can make a direct connection of, oh, this one that you find in Australia is collected in stockyards, and it's the same species that you find in cattle areas in the southwestern US, which is, you know, and then doing a little bit more you find out that there is transport of material, or used to be anyway. I guess now Australia is very careful about importing non-native species. But in Europe, I think, well, Physalis angulata, one of the annual species, I'd have to look back at my FNA treatment. I think it's worldwide that I've seen specimens. I definitely in outside of the Americas, and that seems to be the most widely distributed species. And then probably about maybe 6 or 7 species that have been introduced into Europe, either inadvertently or on purpose. And I think certainly that edible component is important. Maybe some of it had to do with just inadvertent, with travel, with soil, with those pretty inflated fruiting calyces or whatever. So I'm not really sure about all the introductions, but definitely has American origins and has been introduced worldwide.
Yeah, thanks for that. The reason I asked the question was that one of my colleagues is from Ethiopia, and he said that as a child he collected these fruits, and they were eating them. So I'm wondering if how widely distributed. So that's pretty far range from Mexico. Interesting to understand how they got there.
Yeah, and that could be. I mean, I don't know how Physalis angulata, why that one. That's not one of the ones that's comes out as a standout grown for edible fruits, or inflated fruiting calyces that are ornamental. So I'm not really sure why.
Yeah, I agree. I think that one's also super variable in its morphology, isn't it? Would you say? Have you looked at a lot of them?
I don't think angulata is.
Okay.
Not as much as some of the other species. There's that, there's one that has really narrow, it's been named as a variety, as Lanciofolia, has really narrow leaves that, actually that's one of the things that I went out of my way to look at a population in California that it turned out it was growing in the same field as Acutifolia. Nice. And when I saw because the flowers of the two of them are so different, but when you see them on herbarium sheets, unless the flowers are wide open, you can't tell them apart. And I thought, this isn't real. And so I thought, I'm going to go there, and my husband's family lives in California. And so we were visiting, and I said, let's go up and see if this population is still at the roadside edge, and sure enough it was. And I just stood there and said, what do I do with this information? It was just a highlight of how important field knowledge is. So I just hammered home to me how important it is to know the plants in the field, and just how much disappears or can disappear on an herbarium sheet. So
I have a couple more congratulations. If there's not any more questions, we can go ahead and wrap up for today.
Okay.
I'm sure Janet is more than willing to take questions if you have them later via email. You can let me know if you need her email. Next week is going to be Valerian Midland. He's going to be presenting transcriptomics and genomic analysis, revealing new insights into tomato bacteria wilt resistance. All right, have a great week, everyone.
Thank you, everybody. Thanks again.